Today, we are having Dr. Timos Naskas with us. He is a senior program manager in Vision Science Academy. He actually graduated with a bachelor in optic and optometry from the Technological Educational Institute of Western Greece. He also attended the master in clinical ophthalmology and vision research in Glasgow Caledonian University. Timos was awarded a scholarship from the Macular Society to carry out a PhD research project in Queen's University, Belfast. His PhD assessed the structure-function relationship in age-related macular degeneration through the application of multimodal imaging techniques. His PhD project resulted in the largest sample worldwide in the field of AMD. Timos is also interested in health economic. He was awarded the Master Project Manager title as well as fellowship from the American Academy of Project Management. Dr. Timos, very welcome. I just give the screen to you and we are all waiting.
Hello, good afternoon from my part as well. It's a great pleasure to have the invitation received from the president of the Vision Science Academy, and a great honor to get the chance to talk about part of my research that I carried out in Belfast. So we are still working on this publication. We are still refining some of the data. So part of this research is still unpublished, and although you can still find the whole thesis, if someone is interested, I can provide a link to the whole thesis from the library of the Queen's University in Belfast. So what I will do is I will share my screen and take you to my presentation. I have an issue here. I can't share my screen. It says host disabled participant screen sharing. Okay, can you all see my screen?
Yes, it's very clear.
Oh, okay, that's perfect. Thank you. So what we will be discussing today is the use of the OCT imaging technique in AMD. And we will start by briefly discussing AMD and its causes as well as its risk factors. I will not go much into the molecular pathways that lead to the disease and the pathogenesis models. So this will be mostly a descriptive, a brief description of the causes that lead to the development of the disease. And then I will go on to describe the OCT imaging technique and briefly as well the principles of its function. And afterwards, we will share information about how the OCT technique has contributed into better understanding the disease. And then I will go on to develop the methods that I used to bring up this OCT-based classification system for AMD that I developed for my study, and as well as the results that we had. And also I will discuss briefly by the end some future directions, so how OCT can be combined with artificial intelligence for better detecting and treating AMD and retinal diseases in general. So when I was in Belfast, I remember we were writing a paper and one of the professors urged me not to start with a statement that most papers start with. And that goes by saying that AMD is the leading cause of irreversible visual impairment in the Western world. So indeed, it is a huge problem, and it poses, apart from a burden in one's visual function and their lifestyle and quality of life, it also poses a huge burden in a financial burden in terms of healthcare systems, which need to treat all these people. The disease affects people of 50 years of age and older. And according to the WHO, it ranks as the third cause of visual impairment in the Western world. So the first two causes are cataract and glaucoma, respectively. And the meta-analysis, which a meta-analysis, which was a systematic review and meta-analysis, which was published in 2016, made some projections of how the prevalence of the disease is going to evolve until by the year of 2040. So the estimation for 2020 was 196 million people, with populations of the Western world leading the prevalence. So US, UK, and the EU would have most cases of any stage of the disease, any disease stage. By 2040, this number is expected to rise to 288 million people. And the trend is also expected to change. So Asian populations will take over. At least that's what the estimate says. And personally, I haven't come through a paper which would confirm or suggest a new number for the projections that were made at the time for the year 2020. It is a progressive disease. AMD is a progressive disease. It takes several years to develop. And for this, I'm going to develop further for how we actually detect and how we classify the disease in terms of their causation and the strength that each factor has and plays a role into the development of the disease. And I will develop on this just shortly. And a key thing for AMD is that it has a complex pathogenesis. So we are still understanding the disease, and we can't say that we have put the whole puzzle in place to know the full set of risk factors. So as the term implies, AMD is an age-related disease. So age is the key risk factor that plays a role. So the way that I have put this graph here, its interpretation should be bottom-up rather than top-down. So the older an individual is, the more is the risk for presenting AMD. But whenever in this piece of information we add environmental and genetic factors, the risk keeps increasing more and more. So from an epidemiological point of view, every disease can be briefly, the causes of a disease can be briefly classified as environmental or genetic ones. And the thing is, no disease has a pure genetic component or a pure environmental component. Both have a role, and they both have a strength into causing the disease. A simple example for someone to understand this is that you can imagine walking down the stairs, and if you are 100 and you fall from the stairs. So what happens is, if the cause of this was purely genetic, there might be a defective gene that causes your hip not to be as stable when you walk, and this defective gene causes you not to step as well and as stably and cause a fall from the stairs. But if the stairs were wet, you have no problem with your genes. Your grip from the protective bar can be fine and strong enough. But if the stairs are wet, you might slip from the stairs, and that's purely environmental. Although this is not the case in most of the diseases. And we can also we should also take into account the interaction between genetic components and environmental components. And this is mostly well described from the field of called epigenetics. So epigenetics, in brief, is a field of science that describes the modulations in gene activity that can be passed over through cell divisions, but without involving any mutation in the DNA sequence. So epigenetics can help us explain some relationships between environment and gene factors. And what it does, it actually helps us understand how the environment may include relatively stable changes in the traits or even diseases that can be possibly inheritable and passed over through several generations. And actually the gene environment interaction can help modify AMD susceptibility. So the most frequent environmental causes of AMD are smoking and high-fat diet. And I haven't built on the genetic component of AMD because there is a huge genetic background. The most frequent gene responsible for the disease is the CFH gene, which encodes in the inflammatory pathway. But there are several genes taking place. So in terms of aging, we will focus a bit on aging. Biological aging stems from the body's need to undergo metabolism. So while we age, the body tries to undergo metabolical processes, but then these metabolical processes result in the production of damaging toxins, in which then the body tries to protect itself from. And first of all, it's not protecting itself fully. And then the more it tries to protect itself from the effect of these damaging toxins, the more collateral damage the body does to itself. So the path to pathology is these three steps in a very broad concept. So the body trying to undergo metabolism to protect itself, then it's actually producing more damage to itself, which damage builds up over time, and it leads to pathology. A paper from De Grey in the Journal of Gerontology stated that every disease has 7 damaging events in the whole body. So from these 7 damaging events, we have their ocular analogs, so the eye is no exemption. And the ocular analogs for AMD are drusen formation, which comes with age, the thickening of Bruch's membrane, oxidative stress, and other material gathering at the back of the eye, changes in the cells, such as mitochondrial defects, which in the eye results as mitochondrial DNA damage, and cell loss. So taking all these together, there are several factors that need to come in place, and this brings up 2 more concepts, again, from epidemiology. One is the induction period. So again, if we see a disease as a puzzle, as we have here, the induction period is when one of the pieces of the puzzle comes into play. Then, whenever all the other pieces of the puzzle are in place, disease has developed, but we don't detect it until a clinical examination does, or a patient comes to report symptoms to us. So this second period is called the latent period. So through a clinical examination, and OCT has its importance here, through an ocular examination with the OCT, we can reduce the latent period, but of course we cannot modify the induction period for a disease. And I will go back 1 slide. So drusen formation is the hallmark for AMD. Drusen is the accumulation of extracellular material forming between the Bruch's membrane and the retinal pigment epithelium. And it's the characteristic that most classification systems, well, all of the classification systems which are based on fundus photography take into consideration. It's drusen size. So in terms of classifying the disease, there are several classification systems that are in place. All of them have a common characteristic. So here I'm mentioning the four major classification systems used in epidemiological studies. The REDS actually has one additional one, which is their simplified severity scale. For AMD, it's easier both in interpretation and in application as well. So all of these might be different in the philosophy and how they actually classify an eye to a specific disease stage, but all of them have some common components and characteristics. That is, they all, as I said, take drusen size into consideration, and they are based in fundus photography, which was the technology available when these studies had commenced. And one additional classification system, which was well developed and quite, let's say, famous, is the Beckman clinical classification system. Again, this also takes into consideration drusen size and also uses fundus photography, but it is much simpler in its application. It requires both basic clinical knowledge and clinical equipment as well. And taken from this simplicity of the system, I use this to classify my participants to a disease stage whenever I was grading my images, because you should also be an experienced or a certified grader to use the REDS and the Wisconsin or international classification systems for AMD. So I'm not a certified grader. I was not that experienced at the time. And also time was a bit of pressing. So we use the basic, the Beckman clinical system for AMD. So we will also describe how that works. So the Beckman system recognizes 5 steps for classifying AMD stages. So from the left, we have a healthy eye. There we find no pigmentary changes at the back of the eye, no drusen development. And according to the Beckman system, that's considered that is considered a healthy eye. Then we have what is termed normal aging changes, in which there are some small drusen there, less than 63 microns in diameter, and no pigmentary changes, no pigmentary abnormalities present. So in this approach, we keep an eye on this stage, but it's not, let's say, as it's not an emergency as the subsequent stages in terms of frequency of follow-up. And then we go on to early AMD stage, in which the drusen are quite larger. So in early AMD, we find drusen between 63 to 125 microns in diameter, but with no pigmentary abnormalities. In case we find the same drusen size in this stage, but with pigmentary abnormalities, that immediately translates to intermediate AMD. So either drusen 63 to 125 microns in diameter with pigmentary abnormalities, or drusen larger than 125, again with pigmentary abnormalities, would be considered intermediate AMD stage. And then the final stage that the Beckman system recognizes is the late AMD stage, which is split into what we call otherwise wet and dry AMD. Or so dry AMD would be atrophic AMD, and wet AMD would be choroidal neovascularization, where we have vessels breaking through the intraretinal space and leaking blood or fluid. So a key of discussion in this point would be that in a broad manner, some clinicians would term dry AMD even the earlier stages. So as long as there is no fluid there, even early AMD or intermediate AMD would be considered dry, which I personally disagree a bit, but I can understand why they use this this description for the earlier stages. So we will take a slight break from this, and then we will discuss on what's missing from these classification systems. So we said that All these systems use Drusen size, but is it only Drusen that we need to consider whenever we are assessing the disease? It is not, because in 1990, there was a paper which reported a phenotype called reticular pseudodrusen, or otherwise subretinal drusenoid deposits. The term that we use also depends on the modality that we use to image them. So if the phenotype is detected with fundus photography, that is imaging the X and Y axis, we would use reticular pseudodrusen term. If the modality we use to image the phenotype is a modality that images the XYZ axis, that is OCT, for example, we would use the term subretinal drusenoid deposits. So this phenotype was first reported in 1990, and the place it occupied in that paper was merely a paragraph in the results section and only a few lines in the discussion. Since then, it has evolved, and with increasing research on this specific phenotype, it has developed, and we know that it is a major risk factor for progression to late-stage AMD. And also it has a different pathogenesis compared to drusen. So I said earlier, we find drusen between the RPE and the Brooks membrane, but subretinal drusenoid deposits are found at the level of the RPE. So they are mostly found between the RPE and the photoreceptors inner outer segments. And the problem is that why fundus photography hasn't taken into account this phenotype, because it's not as sensitive into detecting the phenotype. So only 33% sensitivity from fundus photography into detecting them when compared to OCT, which has 99%. So much easier and much earlier detected with OCT. Another thing that we do not take into account is variants of wet AMD. And one of these is polypoidal choroidal vasculopathy, which is always placed under the umbrella of choroidal neovascularization. So whenever you start a PhD, you are required to do, excuse me, you are required to do a literature review. And when I was carrying out mine, I was coming across statements asking for an update or a new way to classify disease and possibly be based in a modality that would reflect the technological evolutions. Because, as I said previously, the fundus photography is based on, was actually the technology back available when these large epidemiological studies had begun. But then we see no update in any technology. We see incorporation of new technological imaging modalities, but we don't see a shift to the primary one that is used in clinical or epidemiological studies. So I was thinking that then, if there is such an expressed need to move forward, why should I still use in my research as a primary modality one that is not sensitive into detecting a key phenotype, as it has proved? So, but my problem was that there was no paper focusing only on these aspects. Until in 2017, Richard Spade published this paper, and he was discussing the need to move forward to classify the disease in terms of modalities used, the way that we consider the disease, the phenotypes we take into consideration, and he specifically discussed for subretinal drusenoid deposits and choroidal vasculopathy to be taken into account. So having this, I went to my supervisors and asked them to do something as a first step into creating a new classification system, which they agreed. And then we went over to develop the OCT scheme, which I'm about to develop shortly. So taken from that, we will shortly discuss about OCT. That is a non-invasive and high-resolution technology, which is based on the interference between an object under examination and the reference signal. So briefly how it works, it produces a beam of light which is splitted, and one is the reference beam and one is the beam that goes to the examination tissue. Whenever they come back though, the two beams are going to have differences in terms of time and amplitude, and the interference created will create a pattern which will result in an image, a cross-sectional image, in vivo cross-sectional image of the tissue under examination, and that can be provided in real time. OCT has also been termed clinical biopsy, and its reflectivity, the reflectivity of the layers can be shown in an image. So there's systems which use colors, and hotter colors mean stronger reflectivity, like red and white colors mean stronger reflectivity, and cooler ones like bluish and black colors would mean weaker reflectivity. There are also systems which use different shades of gray, and an image can result in these black and white at grayscale images can result in 256 shades of gray. So I said earlier that the two beams coming back to form an image, if they have differences in time, I can produce an image that is the principle of operation of time domain OCT. And whenever the spectral domain, which is the second another philosophy of operation of the device, uses spectral interferometry combined with a mathematical function called Fourier transformation. And the newer evolution of the spectral domain OCT is the sweat source OCT. It's like a newer version of it. So again, we can use OCT as a clinical biopsy. So several pathogenesis models of AMD, which have been built so far, can be described better, supported, or changed as needed. And OCT has contributed also into detect disease biomarkers earlier, and more clinical trials use it as its primary imaging modality. And also we can use OCT to apply precision medicine into our patients. And now research has taken a step forward into describing and understanding the anatomical features of the retina, with the OCT again being the primary modality. And some studies have taken this even forward. So they combine the results from the OCT, the structural results that we can acquire, with functional results from different tests, such as dark adaptation or microperimetry, which is widely used, which both of them are widely used in the field of AMD, and bring them together so as to have a structure function assessment in the presence of the disease. And as I said earlier, the field of research still formally recognizes the need to develop an OCT grading system for AMD. So there is a good field and a good place to develop this, since there are many research groups working on this. A very important and famous research group is also Cynthia Stoff, which works a lot on OCT and AMD as well. And I think they are now trying to do some, they are in the way of applying a new classification system, but they are still holding back on some publications from that. So in order to build my classification system, what I did was I took the CFP images, first of the fundus photography images from my patients, graded them according to the Beckman classification system. And then I kept that classification there until I developed the OCT as well. And to do that, we followed the narrative literature review approach. And some were considering the narrative review approach not as strong, let's say, or not as accurate as the systematic one. But this was preferred because the field of application And the question we were seeking to answer was quite wide. So whenever you have a wide field of research, the narrative literature view is mostly preferred. And that's why I used mine as well. So we found some studies assessing the characteristics of the retina with using the OCT, and saw what variables they took into account in order to develop our image grading protocol. And then we used the Apostel recommendations to build our protocol and to report our results. And these recommendations were suggested so as every study that uses OCT, their different results are reported in a coherent manner. And I use the Spectralis platform both to acquire and grade my images. So once we finished I finished the literature review, I went on to discuss with my supervisors what items to include in this grading form and how to proceed with that. And I will go through these items and develop also the groups which resulted from this protocol. So first of all, we went on to describe which was the foveal scan. So we described the foveal scan as the one which, sorry, someone is painting over the presentation here. Okay, so the first thing that we took into account was the number of the foveal scan, and that was deemed as the scan that the foveal dip was maximal. And what actually helped us later on to define and determine that point was a small point of hyperreflectivity always present at the foveal dip, at the center of the fovea. And so whatever characteristic we noted in terms of fovea, in terms of the scans found in the OCT image, was then translated into distance from the fovea. So then we described drusen and their metrics. Whenever drus was present, one or more drusen were present, we went on to measure in the largest drusen, its base diameter, its height, as well as the internal reflectivity. Whether that be, so if it would be the same across under the bridge, let's say, of the drusen, it would be homogeneous. But if reflectivity would change throughout, that would be heterogeneous. And that the reason we noted this was that different reflectivities might indicate different materials under the drus. Another thing was subretinal drusenoid deposits. So in the top part of the image, where there's a graphical depiction, so line A corresponds to the inner outer segments of the photoreceptors. Line B corresponds to the interdigitation zone between the photoreceptor outer segments and the apical processes of the RPE. And line C would translate to the RPE layer. And there is a correspective image of the OCT as well to better understand the anatomy. So we did not note only the presence of drusenoid deposits. We also defined their stage. So this is based from a paper by the research group of Sandrine Zweifel, and she recognized 3 stages for drusenoid deposits. So in stage 1, there is just accumulation of granular materials, small mountains at the level of the RPE, which is not sufficient to change the surface of the inner outer segment layer. And then in stage 2, we found this material growing up, and they do change now the inner segment surface, the inner outer segment of the photoreceptor surface, sorry. And then in stage 3, this material has grown, and it's actually pinpointing and breaking through the inner outer segments of the photoreceptors. And there's a 4th stage as well in drusenoid deposits, but the problem is that in order to recognize and determine whether that stage is present, you need to follow up your patients, because what happens in stage 4, there is reabsorption of the material here and eventually some migration towards the inner retina. And next thing we took into consideration was what I termed RP abnormalities. So I remember grading my images and I was finding material there at the level of the RPE mostly, which I could not attribute to drusen. I could not attribute to subretinal drusenoid deposits, but it was clearly something going wrong that was disrupting the continuity and the normality of the RPE layer. So I gave this broad term as RPE abnormalities and included this as a specific phenotype. And also this was translated into a group within the grading scheme later. So in most cases, what was found is just this hyperreflective material getting separated from the RPE layer and traveling towards the inner retina. And another thing we took into account was the thickness of the outer nuclear layer. So the thing with this is that of course, whenever you have material, they are pushing through, and the outer nuclear layer is getting thinner and thinner, as I've pointed with some measurements here. So whenever there's material like a druse in this case, or in this case, the nuclear layer becomes thinner compared to material-free areas. And in the case of geographic atrophy, for example, whenever the disease has evolved and has progressed enough, the nuclear layer would fall off. So then my logic was, whenever I finished and was forming my groups, my logic was that from these factors that I found and these phenotypes that I found, the more material I would find in an image, the higher up would be in the hierarchy. So I have a few examples of the groups. So in group A, there was this image here was considered healthy, and it was placed in the control group. In image B, there is some, I don't know if you can see it clearly, but in this area here, I found some drusenoid deposits, and here as well. And in this case, in this area here, they were stage 2. So this was my SDD group. In the C, in image C, I found both drusen, stage 3 drusenoid deposits and stage 2 drusenoid deposits. So presence of drusen is what defined AMD. AMD was defined if having primarily drusen plus whatever other factors and phenotypes I found. So in this case, where I had drusen and SDD, it would go into group number 6, sorry. So group number 6 goes with drusen along with SDD. And in the case of image D, there's just drusen there, so it would go to group number 5. And EFG translates into my RP abnormalities group. And again, there's clearly material gathering at the level of the RPE or getting separated from it, but still these, whatever is going on there cannot be attributed to drusen or drusenoid deposits. So that was a group by itself. And my late AMD case is in I, there is geographic atrophy, which translates into signal hypertransmission from the choroid. And in the case of image H, there is wet AMD. So again, this, the way that this system was built might not translate what actually happened, might not always translate in a more severe case, but it was based on my logic that the less phenotypes you find at the back of the eye might indicate a less severe case, while the more phenotypes you find might indicate a more progressive case. So then we went on to describe our results. But before I do that, sorry, we also took into consideration the presence of different macular conditions, such as macular hole or vitreomacular traction, and the periretinal membrane as well. And in order to classify, we took a person level analysis and an eye level analysis. So to go to the person level analysis, what we did in both the fundus grading and the OCT grading is we took the stage of the more advanced eye. So in the case of fundus photography, if one eye had intermediate AMD and the other eye had early AMD, that person was assigned the intermediate AMD status. And in terms of statistical approach, we used descriptive statistics. And to compare categorical variables, we used the chi-square test and one-way ANOVA to compare continuous variables. And whenever we compared the two systems, the photographic one along with the OCT system, we used the chi-square test along with the estimation of the kappa value so that we can see whether these two systems would agree with each other. So as per fundus photography, we had the demographics show that there were more females in our study rather than males. And whenever age was compared for the AMD groups, after grouping early and intermediate stages, these participants were significantly older compared to the control group. Also, we found most cases being in the person level, always most cases being in the control and normal and early AMD groups. So we were focusing on the earlier stages of the disease. That's why most people were found at this stage. And then the OCT, we resulted into 7 groups. So there was quite some variance in the prevalence of people in each stage and into what I termed and found to be more advanced stages. Those advanced stages were significantly older than the previous ones. And again, this is where we did not find like a linear change in groups. So the prevalence in each group was varying. It has the different peaks. So that showed us that depending on which group you're focusing, you don't find increasing or decreasing prevalence from one severity group to another. But the key table and the key finding is the one where we describe not only all the OCT features, in which in terms of prevalence of presence of different phenotypes, we agree with previous research, but the most striking finding wasn't the one where we actually compared the fundus photography system with the OCT one. So in general, there is agreement into what we consider healthy with both systems, as well as with what we consider late AMD. But then whenever we go into the other parts of the table, we can see that what we consider healthy with one modality, so what is considered healthy with fundus photography is not the case with late AMD in OCT. So there are things going on at the back of the eye which we cannot detect with fundus photography, but clearly OCT, whenever it's employed, it gives key information. And it can also show, so in what is termed normal aging changes, there are still many biomarkers that are developing, and clearly what is considered healthy in one system is not considered healthy in another system. And whenever we compare the agreement between the two, we found that they only slightly agree with each other. So I have also a small test to see, and if someone could answer, if we have any students in the group, they could answer in the chat box if they would consider this eye here as a healthy one. So if this fundus photography that we have available shows us a healthy eye, or you have an opposite idea. Okay, so I have two answers, and both of them agreeing that the eye looks healthy, and it does look healthy. This fundus photography does look healthy, but then whenever you go for the OCT in two different scans, I found that there's some drusen there. Now, one might bring up an argument that these are just tiny drusen, they're small, they're not merit our attention, but, and that's an interesting debate, but the thing is, it's not normal, first of all, and then how would you give the same weight of importance, and how would you consider these different patients? So it brings us back to what is considered normal with one modality is not actually the case with another one. And sorry for that. So, and again, we have the chance to detect with OCT disease biomarkers much earlier than we do with fundus photography. Yep. And yep, I will take you to the conclusions of this. So this was our first study to have created an OCT-based grading system for AMD. Of course, I did not reinvent the wheel, it's just a first step towards that. And the prevalence of each stage, the way we saw it, was not linear. So that means that whenever you move from a less severe to a more severe stage, you should also have increasing risk for developing late stage AMD. And this part of my research is not put in this table, but I can say that we did not find this increasing risk when we were moving from one stage to another. Although what added to the validity of the way and the philosophy behind this system was that items that we included in our grading form and the variables that we took into consideration were also published in the E3 OCT grading guideline for AMD, which was published 3 months after our grading form was developed. Another thing that merits attention and some further research might be interesting on that is what I termed the RP abnormalities. So we don't know what that is, but we suspected back at the time that these material detaching from the level of the RPE, what it might reflect is that there are RPE cells there which migrate towards the inner retina in order to find alternative nutritional sources. And we also found that, of course, several disease stages and processes are detected earlier. Although we cannot now say that we can change one imaging modality for another. We cannot say that. So one imaging modality cannot exchange for another because we live in an era of multimodal retinal imaging. Several devices offer different imaging capabilities, and each imaging modality can also convey different information in terms of pathological processes going on in the retina. So that would close the part of the research. And in terms of looking forward, there are several AI techniques to localize and quantify pathological features in almost every macular and retinal disease, not only AMD. So these techniques would mimic the way that the human brain works in order to recognize objects, and through learning of more and more pathological features that we found, and they are also very adaptable. So there are different analysis strategies as well in order to estimate the dysfunction and structure abnormalities of photoreceptors in vascular AMD as well as in dry AMD. And there have been also all these algorithms tested can seem to accurately predict how the disease will evolve. But in the end, it is the human brain that is the one to decide how each patient will be treated. So in the end, no matter how much technology has evolved, it will never take the place and should not be never let to take the place of a human in order to take decisions and make, most importantly, clinical decisions, especially in more advanced disease stages. So that would close my presentation. I would like to thank all my study participants, my supervisors, the Vision Science Academy for the invitation, family and friends for their support. And I would like to disclose that I have no financial or no other form of interest from mentioning clinical equipment earlier. And I am at your disposal for any questions. I saw some questions in the chat box and I will reply on them as well. But in case someone wants to ask a question orally, I am at your disposal.
Thank you so much, Dr. Timos, for your presentation. And we are sorry if you saw some interrupted annotation on the screen. We actually keep this annotation for you to just if you want to mark anything, but unfortunately someone just interrupted. We're sorry for that. So there are some questions coming up in chat box. Would you mind, you want to read it or you want me to read it for you?
No, I'll go through them. I'll go through
All
them.
right.
Sure. So Sunanda is also a past classmate of mine in Glasgow when we were both doing our masters. So she's asking if there are any biomarkers that I would suggest to pick up the AMD through sweat source or spectral domain OCT apart from drusen. So for your first question, Sunanda, yes, I would say that drusenoid deposits, since they have been proven a significant risk factor for AMD, even whenever they are found in alone, with not combined with any other phenotype or biomarker, I would pick that up. But again, what changes, as in the example of the small drusen that I showed in the OCT, is how much, let's say, not how much attention, but how you will perceive that. So if I find a few stage 1 subretinal drusenoid deposits, I would not urge a patient to come back earlier than in a year for their follow-up exam. So of course, it's an important biomarker to pick up, but how anyone would perceive and treat it depends on their clinical decisions and the knowledge of the patient's history. So the next question from Sunanda is if there is any correlation between multifocal ERG and OCT, which can give us prognosis of any disease. So I don't quite understand this. So if Sunanda would like to speak and express this question.
Hi, Timos. It was nice to again hear you. Yeah. So the query was like through the multifocal ERG, yes, we can pick up the functional abnormalities of the macula, as we know, and also the OCT also give us the same information about the how the disease has been progressed or the spreaded. So is there any correlation like seeing what we are on the multifocal ERG and what we are seeing on the OCT? Is there anything which could give us the prognosis about how the vision would be preserved or the macula could be preserved, or what is the damage over there? Is there any chances of the giving like a prognosis on based on this too?
Okay, so multifocal ERG can give us information about the functional aspect, and OCT can give us information of the structural aspect. So from my experience, I've done structure-function relationships and correlation with other functional tests. I haven't used multifocal ERGs. I found some correlation between the structure of retinal layers in the presence of health and disease with some functional tests. So I guess, but please forgive my ignorance on this. I guess there might be some correlations whenever you use ERG, but I can't answer this with total certainty because I haven't used it myself. So I guess since you find correlation in one functional aspect with structure, you might also find it when using another functional aspect. So let's say microperimetry that I've used is a functional aspect. ERG is another one. So since you find correlation with one technique, you might expect to find it with another one, but I have some reservations on this. And again, sorry for my ignorance for this, and I hope my answer would be sufficient.
No, thank you. Yeah, we got it. We might need to do some kind of, obviously, the research. Other than it's very hard to give the prediction. So that was just the query, that functional and structural. Anyways, it was a nice presentation, Timos.
Thank you.
Nice. Thank you.
So another participant is asking what are the features that OCT did not pick up, but did pick up the OCT, but not the CFP. So it was not just one feature. We found that OCT can detect drusen, and I chose this small one in purpose to have a debate as well, but there were how to say, larger drusen present. There were drusenoid deposits in which CFP failed to detect them, but with OCT they were clear. Now most drusen deposits, even whenever they picked with OCT, stage 2 and 3 are quite easily detected, but stage 1, because there is no clear, if I can share my screen for a bit, just to show you on this. So whenever I was describing subretinal drusenoid deposits, you can see that the graph shows 3 straight lines. But whenever you go to the actual OCT, you see that the interdigitation zone is not as straight as in the graph. So this is normal, but this can be easily misunderstood with this. So to go back to the question, let me check the question. Yes. So there were several features that the OCT did pick up, but the fundus photography failed to do so. And another question is, what is Fourier domain OCT? Is it different from time domain and spectral domain? Yes, so, well, time, I said OCT has two operating features and two operating principles. One is time domain, where the two beams are coming back and the difference in time is what results in an image, but then spectral domain OCT is based on spectral interferometry. So in terms of time, it uses differences. It uses the differences in the spectrum of the two reflected rays, the two returning rays. But they also use the Fourier domain OCT. So Fourier transformation is a set of mathematical functions which results into an image. So swept source OCT is mostly based on this, and it's actually, let's say, a newer version of the spectral domain OCT. And there is now a debate going on whether it's worth shifting in practice, if it's worth shifting from spectral domain to swept source OCT. And another question I have, what type of OCT was used? And how's the quantity? Okay, so the OCT device I used was the Heidelberg Spectralis. We imaged our patients with the standard 30-degree lens. I also applied for my biomarking a wide field OCT lens, which covers 55 degrees area. And we also used OCTA, autofluorescence with the same device, and multicolor imaging with the same device. And the quantitative measurements we received, so this was another part of my thesis, we used the segmentation. What the segmentation does, it calculates the thickness of each retina layers as well as the retina as a whole. So that was the source and how we acquired our quantitative measurements of the retina. Is there any other question that someone?
Yes.
Thank you.
Yes, there is one more question. Gautam is asking how to differentiate homogeneous and heterogeneous reflective layer in OCT scans.
So does this have to do with the Drusen internal reflectivity that I described, or with layers, with specific layers on the retina? If they could clarify this. With the layers, okay. So if I understand this question well, it goes into how you differentiate layers in general. Is that correct? So how you differ? What is the question? Is the question how differentiate one layer from another? Okay. So what happens is whenever the light traveling. So I'll use this picture. So whenever the light travels in a multi-layered tissue like the retina, the light is reflected, absorbed, scattered in each tissue. So whenever it comes back, it has different properties, and it comes back, let's say, different than what it left the light source, if I can term it that way. So the layers of the retina actually help us understand which layer we are assessing at the time. So you can see that each layer has different reflectivity. So it starts by being hyperreflective, hyperreflective, hyper, hypo, hyper, hypo. And that's what actually drives us. So in a description, excuse me. So in a description, you can see that the first hyperreflective layer, this is a more clear OCT image, and I'll make it slightly larger to focus in. If someone, let's say, starts assessing an OCT image, it's quite easy to navigate through the layers. So the first layer, the nerve fiber layer, is a hyperreflective one. And then the next one, which is the ganglion cell, is slightly hyporeflective. And then each layer goes on into a different manner. So the alternating reflectivity of each layer takes you through the assessment of the retina by, let's say, by itself. So the only thing is the outer retina, so the part of the photoreceptors and the RPE, which needs to be well distinguished, and also Bruch's membrane, so which is at the very bottom, and it's a very thin, it's a single layer, and it's very thin. So that's where some attention needs to go. I don't know if I've covered you with my answer. Is that understandable?
Dr. Timos, we appreciate if you put your email address in the chat box, so if
Yes,
anyone
sure.
gets offered, thank you.
Sure.
Any more questions? Okay, it seems it's yeah, there is no more question. Dr. Timos, thank you so much for joining us.
Thank you very much.
And we just look forward to have more presentation with you. And yeah, people are just saying thank you from Vision Science Academy also. And we are also happy to have you in our team.
Thank you very
And
much.
thank you everyone to just participate in today's webinar. I just take a snapshot. All right. Have a nice day.
Thank you. Have a nice afternoon, everyone.
Bye, everyone.